Hello everyone. In today's lecture, we will continue talking about oxidation of fatty acids, and in this final lecture, we will talk about ketone bodies. Ketone bodies are basically produced in unfavorable conditions when glucose is absent, so that acetyl-CoA, which is synthesized from beta oxidation, that is used for synthesis of ketone bodies. And in today's lecture, we'll talk about how ketone bodies are synthesized and how their synthesis is regulated in a cell. Before that, you need to understand very well about all the reactions of glycolysis, TCA cycle, and beta oxidation. If you remember them, it will be easy to understand everything about ketone bodies. Ketone bodies are basically three compounds which are present in high concentration in blood in case when glucose is missing, glucose is absent, or glucose is in very low concentration in the cell. The first ketone bodies is acetoacetate. This is also called as primary ketone body, and this is converted into acetone by decarboxylation reaction and also converted into beta-hydroxybutyrate by reduction of acetoacetate. So acetone and beta-hydroxybutyrate, they are called secondary ketone bodies. So these ketone bodies, the name is actually historical artifact. Bodies generally means insoluble stuff, but these three compounds which form ketone bodies, they are very much soluble in aqueous environment. In the cell, they are very much soluble, and synthesis of these ketone bodies occurs in liver cells from acetyl-CoA, and this process is called ketogenesis. And they are produced when there is a low food intake, for example in case of fasting, or carbohydrate restrictive diets. A diet is taken where carbohydrate amount is little or nil, during starvation, prolonged intense exercise, and untreated type 1 diabetes mellitus. So in these states, which are not normal physiological states, acetyl-CoA produced in cell is converted into these three compounds. These are called ketone bodies, and after synthesis of ketone bodies in liver cells, they are transported to extrahepatic other cells, and where they are absorbed into the cells and used for energy production. In fact, in brain prefer glucose as a primary source, but beside brain, ketone bodies are the one which are used for production of energy when glucose is not available. In fact, in case of brain, fatty acid cannot cross blood-brain barrier. Only ketone bodies can do that. That's why if glucose is not available, brain survives on ketone bodies only to get energy. And these ketone bodies, they have characteristic smell, the fruity smell they have. For example, acetone is very volatile also. When these ketone bodies present in high concentration in blood, the breath of the person that smell like a fruity smell is there in the breath, and that's one way of detection of high concentration of ketone bodies in the blood of an individual. Acetone is produced in a small amount, is around 2% of all the ketone bodies, and moreover this is volatile, so most of the fraction of acetone that is evaporated from the body. Acetone is known to be chemically inert molecule, but in some studies it has been found out that acetone can be converted into lactate, pyruvate, and eventually to glucose. But because acetone is present in very low concentration, so the glucose produced from acetone is too little to suffice need of energy for a cell. And these ketone bodies, they are basically product of imbalance in carbohydrate metabolism and fat metabolism that we will see in the due slides. Carbohydrates are essential for fatty acid metabolism. If carbohydrates are present, acetyl-coenzyme A, which is produced from beta oxidation of fatty acid, that give energy, that basically enters into TCA cycle so that energy can be produced. But during starvation or in diabetes, where glucose is not present, in that case acetyl-coenzyme A, which is a product of beta oxidation, cannot enter into TCA cycle, so it cannot give energy. So those high concentration of acetyl-coenzyme A in those abnormal physiological states where glucose is absent or in very low concentration, acetyl-coenzyme A is converted into ketone bodies. So, glucose is converted to pyruvate that we called glycolysis. We have talked about this pathway earlier. And this pyruvate is converted into acetyl-coenzyme A, and this pyruvate is also used to convert to have synthesis of oxaloacetate, which is an intermediate of TCA cycle or Krebs cycle or citric acid cycle. We have talked about citric acid cycle in the earlier lecture. Similarly, gluconeogenic substrate, for example, there are some amino acids or other compounds, they are also converted into pyruvate, and from there they can enter into TCA cycle. In another pathway, fatty acid, they are converted, oxidized into acetyl-coenzyme A. This is called beta oxidation. We have talked about this in earlier lecture. So fatty acids are also converted into acetyl-coenzyme A, and they can enter into citric acid cycle to give rise energy. But when glucose is present in low concentration, in that case cells synthesize oxaloacetate. In case of glucose present in low concentration, in that situation gluconeogenesis is promoted. In that, oxaloacetate is converted into phosphoenol pyruvate and finally to glucose. So it means oxaloacetate is depleted when glucose is absent in cell. And when oxaloacetate is not there, TCA cycle, Krebs cycle cannot continue, because in order to have continued Krebs cycle, acetyl-coenzyme A should react with oxaloacetate to form citric acid, to form citrate. But in case of a low carb diet or missing glucose in cell, oxaloacetate is depleted, so that oxaloacetate cannot react with acetyl-coenzyme A to enter into citric acid cycle. So in that situation, accumulated acetyl-coenzyme A in mitochondria matrix of hepatocytes in liver, acetyl-coenzyme A is used in synthesis of acetoacetyl-coenzyme A, acetone and beta-hydroxybutyrate. So these are ketone bodies, and this process is called ketogenesis. So ketogenesis is basically a process which occurs when glucose is absent in cell. So it means the acetyl-coenzyme A, which is coming from beta oxidation of fatty acids, in order to enter into TCA cycle and to give energy, oxaloacetate should be there. But when glucose is low, this oxaloacetate is used in synthesis of glucose, so it is depleted, and that's why acetyl-coenzyme A enter into another pathway for ketogenesis. Liver cell, they synthesize ketone bodies. So free fatty acids, when they are increased in blood, they go to liver and converted into acetyl-coenzyme A in the mitochondrial matrix of hepatocytes. And from there, if glucose is not available, acetyl-coenzyme A, it is used for synthesis of ketone bodies. These ketone bodies from liver, they are sent to blood. Inside blood, they are excreted into urine, that's called ketonuria, or acetone, that is evaporated in breath, and from blood these ketone bodies, they go to extrahepatic other tissues. Other tissues use ketone bodies to produce acetyl coenzyme A, and acetyl coenzyme A enter into TCA cycle to give rise to energy in form of ATP. So that's how extrahepatic cells, they get energy from fatty acid oxidation when glucose is absent. Consequences of ketogenesis. The first thing is formation of soluble permeable ketone bodies. When acetyl coenzyme A in mitochondrial matrix is not being used, so in that case acetyl coenzyme A as such cannot be transported out of the mitochondrial matrix and to other cells. So formation of ketone bodies ensure that acetyl coenzyme A can be circulated to other cells also for utilization in energy production. And ketone bodies are major source of energy for vital organs in case of emergency. Emergency means when there is a glucose concentration is low, like in case of fasting, starvation, or diabetes. And when there is a high concentration of acetyl coenzyme A in mitochondria, so coenzyme A concentration is decreased in mitochondrial matrix, so beta oxidation cannot continue. So in order to release coenzyme A from acetyl coenzyme A, ketone body formation is promoted, because when ketone body is formed, in that case there is a condensation of acetyl coenzyme A molecule, they release some of the coenzyme A which can be used for beta oxidation of further reactions of oxidation of fatty acids. So beta oxidation can only continue if coenzyme A is available. So that's why ketone body formation also release coenzyme A which can be used in further reactions. Transportation, acetyl group, these acetyl coenzyme A cannot be transported. Transportation only occurs in form of ketone bodies from mitochondrial matrix to the cytosol, and from cytosol to liver, to blood and other tissues. In early stage of starvation, heart, skeletal muscles, they consume ketone bodies for energy production, and in that they actually preserve glucose for brain. That happens in case of early starvation. And later, in late stage of starvation, ketone bodies, they are capable of crossing blood-brain barrier. Brain also utilize them for energy production when glucose is missing. Two of the ketone bodies are acid, and that's why when high concentration of ketone bodies present in blood, they acidify blood pH goes down. That process is called, that physiological state is called ketoacidosis. And of course high ketone bodies in the blood, they give a sweet smell of acetone in breath of the individual. Overall accumulation in urine and blood of ketone bodies is called ketosis. And generally in case of high ketone body, in case of high concentration of ketone bodies in blood, give rise to more than 70 mg per deciliter concentration of these ketone bodies into urine, and that is called ketonuria. Ketogenesis is conversion of long chain fatty acid to 3 ketone bodies called acetoacetate, hydroxybutyrate and acetone. And this process occurs in hepatocytes. Acetoacetate is a primary ketone bodies, other two are synthesized from acetoacetate only. Ketogenesis is occur in mitochondrial matrix of the liver cell, and all the enzymes which are responsible for ketone body formation, they are present in the matrix of mitochondria. And the primary utility of ketogenesis is to provide fuel for energy production. This is an adaptive response in case of starvation or in a pathological state when glucose is missing in the cell. Brain oxidize ketone bodies, but fats cannot travel, they cannot cross blood-brain barrier, so those fat in blood, they cannot be utilized by brain to give energy. Other tissues, they can oxidize ketone bodies and fat both. In fact, RBC cannot do that. In case of RBC, mitochondria is missing, so RBC, they solely depend on anaerobic oxidation of glucose. So in that case, ketone bodies also cannot be used by RBCs. Liver synthesize ketone bodies, and the first reaction is condensation reaction of two molecules of acetyl coenzyme A, and that is catalyzed by enzyme called thiolase. Thiolase condense two acetyl coenzyme A molecules and form acetoacetyl coenzyme A. In this reaction, one coenzyme A is released in the cell. This can be used for beta oxidation. Now this acetoacetyl coenzyme A, it is used for synthesis of beta-hydroxybutyrate coenzyme A, in short HMG coenzyme A, where a third molecule of acetyl coenzyme A react with acetoacetyl coenzyme A and form HMG coenzyme A. And same time another molecule of coenzyme A is also released. And this reaction also use one water molecule. This reaction is catalyzed by enzyme called HMG coenzyme A synthase. So this HMG coenzyme A is lysed into acetoacetate. So one acetyl coenzyme A is released in the reaction, which is catalyzed by another enzyme HMG coenzyme A lyase. The first reaction catalyzed by thiolase is the only reversible reaction so far. In fact, thiolase it's also used for splitting acetyl coenzyme A into two acetyl coenzyme A molecule, acetoacetyl coenzyme A into two acetyl coenzyme A molecule also, when ketone bodies are converted into acetyl coenzyme A. So now this acetoacetate, it can be decarboxylated spontaneously or with the help of enzyme acetoacetate decarboxylase to synthesize one molecule of acetone. And acetoacetate is also can be reduced with the help of one molecule of NADH and H+ into beta-hydroxybutyrate by enzyme called beta-hydroxybutyrate dehydrogenase. So this acetoacetate, this is called primary ketone body because this is a parent compound for synthesis of acetone and beta-hydroxybutyrate, and that's why acetone and beta-hydroxybutyrate, they are also called secondary ketone bodies. Now these ketone bodies, after synthesis in liver, they are transported to blood. From blood they go to different tissues, and those tissues capture them. They are ketone bodies transporter on the cell membrane, which facilitate transport of these bodies from blood to the cell, and inside cell, which require energy, and that to be taken from these ketone bodies. They convert ketone bodies into a 2-acetyl coenzyme A molecule. From beta-hydroxybutyrate, that is converted back to acetoacetate. That's a reversible reaction for synthesis also, catalyzed by beta-hydroxybutyrate dehydrogenase, and one NAD+ molecule is converted back to NADH and H+ here. Now this acetoacetate, it uses coenzyme A from one of the intermediate of citric acid cycle, succinyl coenzyme A, and convert succinyl coenzyme A into succinate, and coenzyme A is transferred on acetoacetate to convert into acetoacetyl coenzyme A. This reaction is catalyzed by enzyme beta-ketoacyl coenzyme A transferase, and that also called thiophorase. Now this acetoacetyl coenzyme A is converted into two molecules of acetyl coenzyme A by enzyme thiolase, the same enzyme which catalyze condensation of acetyl coenzyme A molecule into acetoacetyl coenzyme A during synthesis of ketone bodies. Liver cell, they do not have thiophorase enzyme, so liver they can only synthesize ketone bodies. They cannot use, liver cells cannot use ketone bodies for energy production. Similarly, RBC lack mitochondria, so RBC also, they cannot use ketone bodies for the production of energy. Rest other cells, heart muscles, skeletal muscles, kidney and brain, they are all capable of using ketone bodies for energy purpose. So they follow these three reactions to convert ketone bodies into acetyl-coenzyme A molecule. This acetyl-coenzyme A, it can enter into TCA cycle, and that can give rise to, eventually give rise to energy. Now the regulation of ketone body synthesis, availability of substrate long chain fatty acids, and if there are long chain fatty acids are available, they will go for beta oxidation, produce acetyl-coenzyme A, which will be used for synthesis of ketone bodies. Or in case of high fat and low carbs diet, also ketone bodies formation increase, because in low carbs diet glucose is absent, glucose in low concentration, that promote oxidation of fatty acids using beta oxidation reactions. We understood from the previous lectures how glucagon induce beta oxidation reactions. And after beta oxidation means high concentration of acetyl-coenzyme A will be produced, that will favor ketogenesis. The level of malonyl coenzyme A in liver with its influence to inhibit carnitine palmitoyl transferase CPT1, this we have talked in earlier lecture about regulation of beta oxidation. If malonyl coenzyme A is present in liver, that's an inhibitor of carnitine palmitoyl transferase, this enzyme which help transporting fatty acid from cytosol to mitochondria. Then partition of acetyl-coenzyme A between pathway of ketogenesis and citric acid cycle by oxaloacetate concentration. If oxaloacetate is present in good concentration, that favors use of acetyl-coenzyme A in citric acid cycle, but if oxaloacetate is present in low concentration, that favors ketogenesis. And glucagon inhibit glycolysis, increase glycogenesis, and also increase lipolysis, and eventually means glucagon favors ketogenesis. And it's not only glucagon, basically it's a ratio of glucagon and insulin. If glucagon is high and insulin is low, in that case fatty acid oxidation is increased, glucose synthesis is increased, and ketogenesis is promoted. Whereas if insulin is present in high concentration, ketogenesis is inhibited, with beta oxidation of fatty acid is also inhibited. Those we have discussed in detail in earlier lecture of regulation of beta oxidation reactions. So regulation of ketogenesis can be at three levels. In level 1 is lipolysis. Free fatty acids are the precursor of ketone bodies, because we know that free fatty acids are oxidized to acetyl-coenzyme A, and those acetyl-coenzyme A are used for ketogenesis. And factors regulating mobilization of fatty acid from adipose tissue, they control ketogenesis. That also we have seen in detail in earlier lecture, how glucagon regulate mobilization of fatty acids from adipocytes, so that promote ketogenesis. And similarly, in contrast, insulin, if insulin is present, that inhibit fatty acid oxidation, that inhibit lipolysis, whereas glucagon is promote lipolysis, and that's how glucagon promote ketogenesis, whereas insulin, it inhibit ketogenesis. Second level is entry of fatty acid to mitochondria, because if fatty acid are not able to enter to mitochondrial matrix, there will not be any beta oxidation, there will not be any coenzyme, acetyl-coenzyme production in matrix of mitochondria, and hence ketogenesis will not be there. So at this level, the mobilized fatty acid enter to mitochondria for beta oxidation, and we have seen in earlier lecture that CAT1 or CPT1, they regulate entry of fatty acid from cytosol to mitochondria, and they are inhibited by a compound called malonyl coenzyme A. That's a regulator of transportation of fatty acid from cytosol to mitochondria. In case of diabetes or starvation, glucagon is increased, and glucagon it decreases malonyl coenzyme A, and that's how beta oxidation is stimulated. So from this slide we have discussed earlier also, if glucagon is present in cell, that basically deactivate enzyme called ACC, acetyl coenzyme A carboxylase, and when ACC is deactivated, it means malonyl coenzyme A is absent in the cytosol, because ACC convert acetyl coenzyme A to malonyl coenzyme A. That is first step of synthesis of fatty acids. So if malonyl coenzyme A is absent, and that's happening in presence of glucagon, malonyl coenzyme A inhibit carnitine acyl transferase CAT1 or carnitine palmitoyl transferase CPT1. It's same thing. And that's how, if they inhibit this transporter, malonyl coenzyme A inhibit transporter, so that fatty acid cannot be transported from cytosol to mitochondria. So it means glucagon is basically inhibiting synthesis of malonyl coenzyme A, so that CAT and CPT1, they are not inhibited. Therefore fatty acid oxidation can occur. So in short, glucagon is promoting synthesis of acetyl coenzyme A, so that acetyl coenzyme A is converted into ketone bodies, and eventually we can say that glucagon promote ketone body synthesis, ketogenesis. Third level of regulation is oxidation of acetyl coenzyme A, and when the previous two steps they are active, more acetyl coenzyme is produced, because in the previous two steps we have seen that fatty acid, they are able to transport from cytosol to mitochondria, and beta oxidation is increased, and when beta oxidation is increased, acetyl coenzyme A is present in high concentration, and this acetyl coenzyme A is completely oxidized in TCA cycle. So if TCA cycle is active, acetyl coenzyme A will enter into TCA cycle, and eventually from oxidative phosphorylation ATP will be synthesized. But in case of diabetes mellitus and starvation, acetyl coenzyme A cannot enter into TCA cycle, because in starvation and diabetes, oxaloacetate is depleted from the cytosol for gluconeogenesis. That's why acetyl coenzyme A produced in beta oxidation, that's used for synthesis of ketone bodies. So if we summarize all the factors for ketogenesis, HMG coenzyme A synthase, this is an enzyme of ketogenesis. Its activity is induced by increased fatty acids. If fatty acids are present in high concentration in blood, HMG coenzyme A synthase activity is increased. It means ketogenesis is increased. Coenzyme A level also regulate ketogenesis to retain coenzyme A pool in mitochondrial matrix. Reduced coenzyme A level stimulate HMG coenzyme A. So if coenzyme A is present in less concentration in mitochondrial matrix, it means ketone body synthesis should occur, so that some coenzyme A molecule, they are released, which are trapped in form of acetyl coenzyme A. So that's why reduced coenzyme A level stimulate synthesis of ketone bodies, and if coenzyme A is present in high concentration, they inhibit HMG coenzyme A synthase, so that ketone bodies, they are limited, their synthesis is limited. This rate of ketogenesis and its efficiency directly depends on many factors. We can summarize them. Insulin activity, if insulin is present, ketogenesis is inhibited. Level of cellular glucose, if glucose is depleted, only then ketogenesis occurs, otherwise not. Level of oxaloacetate, if oxaloacetate is present in high concentration, acetyl coenzyme A enter into citric acid, so no ketogenesis occurs. high rate of lipolysis and fatty acid oxidation, they also enhance ketogenesis. And underutilization of acetyl coenzyme in citric acid cycle, that also promote ketogenesis. And large accumulated amount of impermeable acetyl coenzyme in mitochondrial matrix, that also enhance ketogenesis. All these factors are interrelated with one another. For example, insulin activity. Insulin is only present when there is a low glucose level concentration. And when there is a low glucose level concentration, means gluconeogenesis will occur, because in low glucose concentration, glucagon is secreted. And when gluconeogenesis is occurring, oxaloacetate is depleted, so that acetyl coenzyme cannot enter into TCA cycle, and hence it enters into alternate pathway, that's called ketogenesis. The rate of synthesis of ketone bodies in liver is such that these synthesized ketone bodies can be easily metabolized into other tissues, and that's why even in normal state ketone bodies concentration is less than 1 mg per dl. But when the rate of synthesis of ketone bodies exceed rate of utilization, for example in case of diabetes, when insulin is missing, glucose is not there and glucagon is present, in that case concentration of these ketone bodies increase in the blood, and that's called ketonemia. So increasing high concentration of ketone bodies in blood is called ketonemia. And after ketonemia, when you have high concentration of ketone bodies in blood, they are excreted in urine also in high concentration, and that's called ketonuria. And at the same time, high concentration of ketone bodies in blood give rise to a smell of acetone in the breath, some fruity smell, and together ketonemia, ketonuria, and this smell of acetone in breath, this is called ketosis. So ketosis is a pathological state where high concentration of the ketone bodies are present in the blood, urine, and breath of the person also smell like presence of these ketone bodies. And together all these symptoms together, all these parameters, they are called ketosis. So we can summarize them. The causes of ketosis: in case of diabetes, mellitus, insulin is present in low concentration, so glucagon is increased. When glucagon increases, it enhances lipolysis. Lipolysis gives rise to high fatty acid concentration in plasma, and these high fatty acid, they enter into liver and hepatic cell. They promote ketogenesis, and more ketone bodies are produced, and that lead to ketoacidosis. Similarly, in case of starvation or fasting state, pancreas secrete glucagon because glucose concentration is low in the blood, and this glucagon activate oxidation of fatty acids. Fatty acids are liberated from and those cells, adipocytes, and these fatty acids go to liver. They are converted into ketone bodies. Ketogenesis is promoted. Ketone bodies enter into blood and transported to different tissues. So in case of overnight fast also ketone bodies are formed, but the concentration of ketone bodies is not too high. In case of overnight fast is 0.1 to 0.4 millimolar. But if fasting is prolonged, like in case of starvation, this concentration can go up to 7 to 10 millimolar. That's very high, and that's lead to ketosis. Ketone bodies can be tested using Rotheras test. That's a classic nitroprusside reaction where acetoacetate react with sodium nitroprusside in alkaline conditions and give a purple color. And this purple color is detected, is measured in order to have estimation of presence of ketone bodies in urine or in blood sample. Another test used 10% ferric chloride solution, and when this is added in a solution like urine, and it's become reddish and purplish in color, and this color is estimated to have some idea about concentration of ketone bodies in the blood. The normal level of ketone bodies is acetoacetate is present in 0.1 mg per 100 ml, which is increased to 7 mg per 100 ml in case of ketosis. Beta-hydroxybutyrate around 8 mg per 100 ml, increased to 30 mg per 100 ml in case of ketosis. Free fatty acids are 9 mg per 100 ml in normal level, and that's go up to 28 mg per 100 ml in case of ketosis.